Okay, in our previous class, we discussed about what are amino acids, definitions, classification, and also we discussed about uh, two methods of synthesis of amino acid. Amino acid, okay, let's give an entry. Okay, so in our previous class, we discussed about what are amino acids, classifications, acidic amino acids, and neutral amino acids. There are 20 amino acids, uh, and their examples. Okay, with the structure and symbol also we discussed, and we also discussed about synthesis of amino acids, starting with an alpha halo acid. Okay, so this is an alpha halo acid. So this alpha halo acid on amination gives ammonium salt, and then hydrolysis of ammonium salt gives amino acid. If we had, okay, let's give an entry. Okay. So first method of synthesis of amino acid is starting with an halo acid. Suppose if we are taking a different halo acid, if we are taking a propyl, 2-bromo, otherwise alpha bromo propionic acid. Okay, if we are taking a here one CH3 here, one CH means isopropyl group. So isopropyl group here and bromo is here, then you will get a valine as the product. So only thing, if we are changing this halo acid, your amino acid also changes. That's what you need to remember. The first method of synthesis of amino acid is with starting with alpha halo acid. So by this method, it is possible to get valine, leucine, aspartic acid, alanine, and glycine. So one more method is from malonic acid. So this is the ethyl malonate. Means malonic acid, when it is esterified ethanol, you will get a ethyl malonate. So this ethyl malonate, on treating with a electrophile. Here, C6H5CH2Cl, benzyl chloride. So base captures this proton, forms an anion. That anion react with this CH2, Cl and H will go out, forming a compound like this. So this compound on hydrolysis, ester is converted into acid. So the and then bromination. So this particular carbon is there. Now it contains proton. That proton is brominated. So the a bromo compound is obtained. So this compound, it is a acid, more particularly having a dicarboxylic acid attached to the same carbon atom. Such compounds are temperature undergoes decarboxylation. So CO2 will go out. So this bond pair picks up this proton. So that is how phenylalanine is obtained. Suppose if you want to prepare a alanine, so then instead of benzyl chloride, if we are taking a methyl iodide, then you will end up with alanine as the product. If you want a valine, so here isopropyl chloride you should take. So isopropyl chloride means CH3CH, CH3Cl. So this will attack CH carbon, and remaining steps in place of benzyl, you can add CH, CH3, CH3. Okay. So there you will end up with valine as the product. So this is how the two methods, how one can synthesize amino acid starting with malonic acid as well as the alpha halo acid. So few more methods also is there. Let's discuss about how the preparation. So I will method of synthesis of amino acid. So this is the Gabriel thalamide synthesis. It's a name reaction. Okay. So here what will happen? A thalamide. Okay. Thalamide means this is the one. So without this NK, K means potassium, N means nitrogen. So without these two, if there is a COH here, then it's called phthalic acid. So phthalic acid when it is, okay, let's give an entry. Okay, so phthalic acid when it is on heating gives phthalic anhydride. So phthalic anhydride on treating with ammonia gives phthalimide. Okay, so this phthalimide, you know that N minus and K plus. So N minus will attack this carbon and Cl will go out. So removal of KCl. And what is the name of this compound? So this position is called alpha position, alpha chloro. Alpha chloro ethyl formate is the trivial name. If you want to look into the IUPAC name, so this is one carbon atom, this is two carbon, 2 chloro ethyl acetate. Okay, 2 chloro ethyl acetate is the IUPAC name. 2 chloroacetate is the common name. Then what will happen? So there will be a new bond formation between this carbon and this carbon. So that's what I have written here. So this compound on hydrolysis, hydrolysis means breakage here. So breakage here. So this N is converted into NH2. So that is how a glycine is obtained. And this is a phthalic acid, it's a byproduct. It will go out. And here one thing you need to remember, by using a Gabriel thalamide method, it is not possible to synthesize phenylalanine. Okay, it is possible to synthesize alanine leucine. Okay, why it is not possible? Because phthalimide cannot, I think nucleophilic substitution at the aromatic ring is not possible. Whereas phenylalanine, of course, it's an aryl alkyl halide. So that is possible to some extent. But uh, uh, using a Gabriel thalamide method, it is not possible to prepare aromatic amines. Okay, it is a method of preparation of amines. Okay, primary amines. It's also a good method for the preparation of amino acids. Okay, so here what is involved? A thalamide is treated with halo esters, forming a obtained compound is on hydrolysis gives amino acid. So if we are taking a 2-chloroethyl acetate as the electrophile, substituted one, hydrolysis means breakage here, breakage here. So this carbon is converted as COOH, this carbon as COOH, this nitrogen as NH2. So glycine. Suppose if we are taking a different, uh, suppose if there is a substituent at this position. Okay, okay, that I will discuss in the next slide. Okay, so I will go to the next slide. So here I am taking a different compound. You just observe. So malonic acid I am taking here. So combination of malonic acid and then phthalic acid. I think there should be a what is called as you cannot take directly malonic acid. Maybe you need to take it as a bromomalonic acid. So then there will be a reaction between phthalimide and bromomalonic acid. Bromo means here. So then there is a reaction between these two and forming a compound like this. So after this also, this carbon is acidic in nature, this proton is acidic in nature. So now also this proton is acidic in nature. So in presence of base, can obstruct this proton, forms an anion. That anion can attack one more what is called halo ethyl chloroacetate. So then there will be a intermediate formation like this. So this on hydrolysis. Hydrolysis means breakage of phthalimide group, converting into phthalic acid and then aspartic acid. So this is how aspartic acid is obtained with melanic with phthalimide method of amino acid synthesis. So here what you need to remember, so phthalimide method of amino acid synthesis means potassium phthalimide is treated with halo ethyl halo acetate, and the obtained product on hydrolysis gives amino acid. Okay, that's what you need to remember. So with this, I will go to the next slide. So next slide, I will discuss one more method of synthesis of amino acid. So it's called a Stecker method. Here what will happen? A reaction of aldehyde with hydrogen cyanide. So what will happen? H plus will add to will form a bond with oxygen, and Cn minus will form a bond with carbon. So the obtained compound is called cyanohydrine. This cyanohydrine, when it is treated with amine, okay, so it replaces the hydroxy group. That means H from ammonia, and OH here goes out as a water, forming amino alcohol. This is amino, this is nitrile. Amino nitrile is formed. This this amino on hydrolysis. I think hydrolysis means here a cyanide is hydrolyzed. So cyanide on hydrolysis, what will happen? Cyanide on hydrolysis gives an amide, later acid. So let's give an entry. Okay, all right. So in this method, when an aldehyde and hydrogen cyanide gives cyanohydrine, this cyanohydrine on treating with ammonia, it replaces hydroxy group, forming an amino nitrile. Amino nitrile. This amino nitrile on hydrolysis. Hydrolysis means CN uh, uh, hydrolyze to carboxylic acid. So here this N will ammonia. So this method of synthesis of amino acid is called Strecker's synthesis. An aldehyde is treated with ACN to form a cyanohydrine, so which is made to react with ammonia to form a aminonitrile. The nitrile on hydrolysis yields alpha amino acids. So here I am taking an example. If you are taking formaldehyde and hydrogen cyanide, you will end up with glycine. If you are taking acetaldehyde and hydrogen cyanide, you will end up with alanine. So by this method, it is possible to synthesize valine, leucine, isoleucine can be synthesized by this method. So this is about Stecker method. So Stecker method means a reaction of aldehyde with hydrogen cyanide gives cyanohydrine. Cyanohydrine on amination gives aminonitrile. Aminonitrile on hydrolysis gives amino acid. So that's what you need to remember with respect to this method of synthesis. So with this, I will go to the next slide. So next slide, I will discuss one more method of synthesis of amino acid, that is Curtius rearrangement. Curtius rearrangement reaction. So I will tell you where is the Curtius rearrangement means. It's a conversion of azide. Look here, this group is called azide. NH3 means azide group. So this azide is converted into carbamate. This carbamate on hydrolysis gives amino acid. The conversion of azide Okay, more particularly acid azide to amide is called Curtius rearrangement. So to get this acid azide, we have to start with alkyl halide. So Rx means alkyl halide, and this one is ethyl malonic ester. So what will happen in presence of base? There will be a anion formation. That anion react with R, and X is going out. So a compound is obtained. Means it's a kind of nucleophilic substitution reaction. So this on hydrolysis, here ester is there, no? Ester is converted into acid. And here, if you are taking a one equivalent, then it is possible to hydrolyze only the one ester group. So that is how a mono carboxylate, mono potassium salt is obtained. This mono potassium salt, when it is treated with hydrazine, so this hydrogen react with this carbonyl compound, and OET. OET means ethoxy group is going out. So that is how hydrozone is obtained. This hydrozone I am treating with nitrous acid. HNO2 means nitrous acid. So it will convert this hydrozone to azide. N3 means azide, and CO means acid azide. So from here to here, if you want to know that, it's a Curtius rearrangement. Curtius rearrangement means here nitrogen will go out, and this particular bond is there. No, this particular bond is migrated from carbonyl carbon to nitrogen. So this is nothing but a uh, migration of electron deficient nitrogen. So migration of electron deficient nitrogen. That means this carbon, this bond is migrated from carbonyl carbon to electron deficient nitrogen. Why it is electron deficient? Because nitrogen is going out. So that is the reason this one is electron deficient. The obtained compound is called carbamate. This carbamate on hydrolysis. Hydrolysis means amide, NHCO is there. No, NH and CO is breaks. So similarly, the bond between the CO and OED, okay, that is also breaks. So here what will happen? Ethanol is one product, carbon dioxide is another product. So this carbamate initially hydrolyzes, a carbamic acid is obtained. Carbamate is converted into carbamic acid when it is treated with a base. Okay, here base is required. So base, that base convert ester into acid. Acid means NHCOOH. That carbamate on decarboxylation, CO2 is going out, forming a free amino acid. So this is about the Curtius reaction involving amino acid synthesis. Say here you can write down where R is equal to H, it's a glycine. Where R is equal to methyl, it's a alanine. Where R is equal to isopropyl, then it is valine. Where R is equal to benzyl means C6H5CHO, then it's phenyl alanine. So, so based on this R, you will get a different amino acid. So, but this reactant is same. So this is about Curtius reaction. So up to this, we discussed about what is called as Stecker synthesis, Curtius rearrangement, and Gabriel thalamide method of synthesis of amino acid. So Gabriel thalamide, Stecker synthesis, and Curtius reactions. So from all these methods, it is possible to synthesize a amino acid. So with this, I will take one more method of synthesis of amino acid. So that is what is called as the ROPC. So here what will happen? An aldehyde is treated with cyano. This is called uh, ethyl cyano. This is ethyl cyanoacetate. ethyl cyanoacetate. Okay, so an aldehyde is treated with ethyl cyanoacetate. What will happen? It's a condensation reaction. Condensation means removal of two hydrogen present here and oxygen goes out as a water, forming a new bond between this carbon and this carbon as a double bond. Double bond CH. Okay, so there will be a formation of double bond. So that double bond is reduced to single bond when it is treated with hydrogenation in presence of palladium. So this is nothing but a condensation followed by hydrogenation. So in this method, aldehyde is condensed with ethyl cyanoacetate and simultaneously hydrogenated. So to get an intermediate like this. So this intermediate, when it is treated with N2H4, N2H4 means hydrazine. So what it will do? It will react with this carbonyl carbon, forming CONH2. It's called hydrazone. So that hydrogen, when it is treated with nitrous acid, it will give you acid azide. So this acid azide on treating with a ethanol in presence of H plus. So what will happen? It's a courteous rearrangement. So there will be a migration of this bond from this carbonyl carbon to nitrogen, forming a carbamate. That carbamate hydrolysis and then decarboxylation and giving rise to amine compound. So this is about the Rapsi method. So the Rapsi method means reaction of an aldehyde with ethyl cyanoacetate. A condensation followed by hydrogenation takes place. The obtained compound is treated with hydrazine to get hydrogen. The hydrogen was treated with nitrous acid to get a acid azide. The acid azide undergoes courteous rearrangement followed by hydrolysis gives amino acid. This is about the Rapsi method of synthesis of amino acid. So one more, the last method is there, that is called azalactone method. So this is called, of course, it's also called Erlenmeyer. I think you are aware about the conical flask. So one more name for the conical flask is called Erlenmeyer flask. So Erlenmeyer is a scientist. So what is happening here? Erlenmeyer azalactone synthesis of amino acid. So by azalactone, it is possible to prepare phenylalanine. Of course, other amino acids like leucine, valine, alanine, glycine can be synthesized by this method. So what is here? You should be aware about epuric acid. Of course, this particular epuric acid you are going to synthesize in a laboratory when you are in the third semester. Okay, so then what is epuric acid? You know that C6H5CO is called benzyl group. C6H5CO is called benzyl group. NHCH2COOH group is called glycine. So a glycine, when it is treated with benzyl chloride, it will give you epuric acid. So this epuric acid, when it is treated with benzaldehyde, so you will get azalactone. So again, as usual, in the previous case, what you observed? You observed that condensation reaction followed by hydrogenation. So here also, there will be a condensation between this carbon and this CHO. So that is how a compound is obtained. Double bond C6H5 is obtained. Then afterwards, what will happen? So condensation, one more condensation. I think you know that this is a acid, this is a ketone. The lone pair present on the oxygen can react with this carbon, forming a OH. This OH and this H, okay, going out as a water and forming a double bond here. So that is how a 5-membered azalactone is obtained. Okay, and here it's only the condensation and cyclization. So there is no reaction of hydrogen in presence of catalyst. This is not required. Okay, this you can erase it. Okay, H2 palladium is not required for the conversion of benzaldehyde and epuric acid to azolactone. Okay, so the obtained azolactone, when it is treated with 1% sodium hydroxide, that means cleavage here. Okay, converting back into amide and then acid. So this was subjected to sodium amalgamation. So sodium amalgamation means reduction of this double bond to single bond. So and then and then cleavage of this protection. Okay, this NH was protected with benzyl group. No, that cleavage it is possible within sodium hydroxide itself. Okay, then the obtained compound is called phenylalanine. So these are the methods of synthesis of amino acids. So the first method is treating with an acid, halo acid. Second method is about the from the malonic acid, malonic acid, how the amino acid is synthesized. And the third method is Gabriel thalamide synthesis. Fourth method is Stecker synthesis. And the fifth method is Curtius rearrangement. And the sixth method is what is called as Darabsi method, and the seventh method is azolactone Erlenmeyer method of synthesis of amino acid. So these are the 7 methods of synthesis of amino acids. Okay, now tell me, is there any doubt in any method of synthesis of amino acid? Any doubt?
No, sir.
Okay. So in this chapter, first we are discussing about what are amino acids, their classification, and the next is about the synthesis of amino acids, and the next is about the peptides, how the peptide is formed. Okay, and after the for peptide nomenclature and how it is formed, so we will discuss about different what are the problems in peptide bond formation. Okay, how one can selectively synthesize stereo specifically required peptide, and what are the different methods available for the peptide synthesis. Okay, that's all about this chapter, but we will go by stepwise, stage by stage. So up to this, we discussed about what are amino acids and how their classification and their synthesis. So now onwards, I will discuss about peptides. What are peptides? So what are the what is the difficulty in synthesizing the peptide? To overcome, what are the systematic methods used to synthesize a desired peptide? And what are the reagents used in peptide synthesis? So those things we will discuss in our next class. So I think you know that. So peptide is nothing but a formation of union of amino acids. Okay, suppose if two amino acids are joined. Okay, I think you know that amino acid means it contains a amino group as well as carboxylic acid group. Suppose a amino group of one amino acid react with the carboxylic group of another amino acid, then the obtained bond is called peptide bond, and the obtained compound is called dipeptide. Okay, so I will go to the next slide. So look here, peptides. So what are peptides? Are the amides. Okay, peptides are the amides. So formed by interaction between amino group of one amino acid with carboxylic group of the other amino acid. And the amide group, okay, the resulting bond is called a peptide bond. Of course, suppose if we are taking a uh, other than amino acid, okay, then that bond is called amide bond. So if we are taking a amino acids, then the formed bond is called peptide bond. And the linkage, NHCO linkage, is called peptide linkage. So when two amino acids combine, the peptide formed is called a dipeptide. So when three amino acids combine, the peptide formed is called tripeptide. When many amino acids combine, the peptide is called polypeptide. Then what are proteins? Proteins are the polypeptide containing at least 100 or more amino acid and having the three-dimensional structure is called the proteins. So just I will repeat. Peptides means how the peptide is formed. Amino group of one amino acid interacts with the carboxylic group of the other amino acid, leads to the formation of the bond called NHCO. The NHCO bond uh, is called peptide linkage. Of course, it's also called as a amide, but it is not in the case of the two amino acids are involved. If the two amino acids are involved in a amide bond formation, that amide bond is called peptide bond. That linkage is called peptide linkage. Okay, this is the important thing you need to remember. Suppose two amino acids are joined, then the obtained one is called dipeptide. If three amino acids are formed, then the obtained one is called tripeptide. If it is a more, tetra, penta, okay, hexa, deca, and then if it is a more, then it's called polypeptide. Suppose proteins are, if you want to say about the protein, means protein is a polypeptide containing more than 100 amino acid units and having a three-dimensional structure. One is called protein. So now we will discuss more about the peptide only. So with this, I will go to the next slide. Here I am taking an example, okay? The peptide bond, how the peptide bond, more particularly nomenclature, I will discuss in the next slide. So look here. So here I am taking this is the NH2, CH2, COOH is called glycine part. So here what is that? Carboxylic end is reacting with the amino end of the phenylalanine. Okay, this is a phenylalanine. So what I can say that now amino group of the phenylalanine react with the carboxylic group of the glycine forming a bond. This bond is called peptide bond. Of course, this linkage also is called peptide linkage. And one more thing, normally peptide is written in such a way that left hand side is N-terminal end, right hand side is C-terminal end. So C-terminal end means it should have a free carboxylic moiety. N-terminal end means it will have a free NH2 group. So normally peptide is written in such a way that left hand side there should be a N-terminal end, right hand side there should be a C-terminal end. If I am taking a dipeptide, it's okay, quite easy. So suppose if there is a what is called 100 amino acid containing one. So then also left side you will have a free amino group containing uh, N-terminal end containing amino acid, C-terminal end containing amino acid. In the left hand side, free amino group will be there. In the right hand side, free carboxylic group will be there. Okay, this is about N terminal and C terminal of the peptide. Okay, uh, right. So maybe I will go to the next slide. So nomenclature. So peptides are named starting from the N-terminal end. So that means if you want to name any peptide, you should come from the left-hand side. And what is, I think you know that glycine, alanine, okay, phenylalanine, all these things end with INE. So when you are naming the N-terminal end, INE should replace with YL. INE should replace with YL. For all amino acid except C terminal. If it is a C terminal means you have to write as such. So in that case, if you want to name this particular dipeptide, so you have to find out what is the N-terminal end. So N-terminal end is NH2CH3CO. So since there is a CH3 means it's called alanine. So in that alanine, INE should replace with YL. So then name of this compound is called alanyl. And this C terminal end is glycine. So you have to write as such. So name of this dipeptide as per the IUPAC name is called alanyl glycine. In terms of symbol, you can write ALA-GLY. ALA stands for alanine, GLY stands for glycine. And here one more thing you need to remember that ALA is the N-terminal end, GLY is the C-terminal end. Suppose a peptide contains 3 amino acids, you have to identify. Of course, since there is a H here, it's a glycine part. Since there is a CH3, it's a alanine part. Since there is a CH2 phenyl, it's phenylalanine part. So then, if you want to name IUPAC name, glycine, INE should replace with YL, glycyl. Alanine, INE should replace with YL, glycine, alanyl. And the C terminal end you have to write as such, phenylalanine. So name of this tripeptide is called glycyl alanyl phenylalanine is the IUPAC name of this tripeptide. And if you want to write in terms of symbol, GLY glycine, ALA alanine, phenyl PHE is the phenylalanine. So this is about the nomenclature and how the dipeptide is formed and the N-terminal end and C-terminal end and 7 methods of preparation of amino acids. Maybe what I will do, okay, up to this, if you have any doubt, you can ask me. Otherwise, uh, maybe in the next class, I will continue the remaining part. If you have any doubt up to this, you can ask. Do you have any doubt? If
No,
there
sir.
are no doubt,
No, sir.
okay, you can log out for today's class. Maybe tomorrow I will take as usual